is that we need to, neurons are very complex structures in terms of their trees. And these ion channels are inserted in different locations. And depending on the locations, this can add new functions to the neurons. So we not only need to know which of the 20 or 30 ion channels they're going to select from the pool, we have to know exactly where to put these ion channels. So for a whole neuron, you're talking about several million ion channels, several types, and you have to know where to put them. So we use many, many different techniques, and I won't go into all of them, but this is freeze fracture scanning microscopy, and you can actually locate single channels, single molecules, to constrain the model and say that there are certain distributions and certain locations for these ion channels. So we think that, and we're in the process, that it will be possible to start putting these ion channels in exact locations. We've recorded from over 10,000 cells, and we've begun to start deriving what we call the first recipe of the neocortical column. The recipe is the recipe of cell types that are used to build up the circuit. Now, as I said, the neocortex from mouse to man, this column is surprisingly stereotypical. There are many, many features, that approximate ratios of excitatory to inhibitory types of cells, the types of interneurons that they are. But the subtle recipe may change across species and across regions, but essentially the core aspect of this recipe stays very much the same to give you a column. So once we have this template, we would be able to begin building columns from different species, different brain regions, and so on. The next thing that we need to be able to do is, of course, this is an infrared picture of the whole column, and this is what it looks like if you just pack in a few of these cells. Red are the dendrites, blue are the axons, but we now have the neurons. We can model their structure, we can model their function, but how are we going to connect them? So we've done a number of different types of experiments, and the first is that what we wanted to know, Jerry Edelman raised an important issue in the neocortex with so many specific things. Is it possible for one neuron to go and target a particular cell? And he's in the past made a very clear claim that it's not possible. So we wanted to check this experimentally, and what we did was to record from two cells, fill them with a dye, and use a confocal microscope to do a very, very detailed three-dimensional reconstruction. And then we take the axon and we track it, we follow it, and we count every time it gets within 0.1 micrometers of the dendrite of a target cell. And then we do statistics to see whether for connected and unconnected cells, there's any bias as to how the branches are going to target their different cells. And as Jerry would have predicted, there is no bias. Every axon touches every target cell. It's an all-to-all circuit that is ready and in place for a specific functional circuit. And we could see that when, because it's not going to transmit information to all of them. It's just structurally in position. It's only going to transfer the information to about 10% of them. And those 10%, all it has to do is grow a synapse. And I'll come back to that at the end and show you that that's a very dynamic process. Now, we know, we use that, and we're going to come back. We're going to use that information about all-to-all structural connectivity with certain numbers of touches on each cell to constrain the way we position these neurons in 3D space when we build the circuit. But we need to know more than that. We need to know for each type of cell, not only that it touches it, but exactly where it touches it. What part of the axon is used, and on what part of the dendrites does it place those synapses. So what we've done is to record from lots of cells that are connected. This is a small basket cell that is connecting 3 pyramidal cells. And the red shows points of information exchange, synapses. It looks like a mess, but if you do this repetitively for the same type of pre- and postsynaptic neuron between a pine tree and an oak tree, you find that there are patterns. They're very specific patterns in terms of which parts of the roots are used to contact which parts of the branches. And this information is the most crucial information to building a biologically accurate circuit. Nobody in the past has bothered to collect this data because it's extremely tedious. But these are the locks and keys that will tell us and allow us to place 10 to 20 million synapses within the size of a pinhead in exactly the right location, so that they will exchange information in the right places. And we're going to use that. So what we then did was to build a circuit builder, and it's an application which interfaces with the database of all these different neurons that we repaired and cloned and functionalized. We enter the recipe. We define the cortical column and the positions of all the different neurons, the first positions of the different neurons, and then we load them. And what you're seeing here, and this is now a hexagon because we're anticipating coupling these columns to build larger parts of the neocortex. But what you're seeing here is the loading of 10,000 neurons, starting in layer 2, 3, layer 1, layer 2, and the blue are the axons and the red are the dendrites. So, as I said, we're packing together in the space approximately, which is about 300 micrometers in diameter, we're trying to pack and position 10,000 neurons. Now, we use other types of rules, such as mini-columnar rules, to give the first initial position. But one's got to be aware that it doesn't help just putting the neurons there. They're going to have to move and jitter and spin in order to get the exact locations that fit the experimental data that says this is where the synapses are. You can see how much spillover there is that is used to connect onto the next column that is sitting side by side. So the next thing we need to do is, once we've positioned them, we've put the neurons first in position, we now need to connect them. And we do two steps. First, we have to structurally position them according to that statistical information that we derived before, and then we have to functionally connect them. So this is just a zoom up of the dendrites in gold and the axons in blue. We have to run a collision detection for every time that the axons touch the dendrites. And they're touching approximately 100 million times. And we do this on the blue gene, so it's quite a challenge because you have pieces of neurons and the axons and dendrites all over 8,000 processors. And it is one of the most demanding challenges, which we're working closely with IBM to work on algorithms to do this faster and faster, because right now we can do just the first iteration, but we need to do 10,000 iterations while we spin and jitter the neurons and check that they are structurally positioned correctly and that they are the locations are exactly according to the lock and key principles that we've derived before. So once we do that, we can now connect these neurons according to statistics of connectivity, which is our initial process. We're going to use algorithms later. But statistics of connectivity says, we've structurally positioned you, you've touched, but only 10% are now going to be connected. And those 10%, when they connect, they will put multiple synapses on each other. And that's how we solve the problem of having random structural connectivity, but specific functional connectivity. And this is more or less what a single neuron looks like, packed with synapses in many different locations, from the tough dendrites to the basal dendrites and around. We can also zoom in on a single cell and color map the source location of the different neurons. Now, as I mentioned to you, we have this lock and keys. The lock and keys translate essentially into a specific color map for every type of cell. So we can use the color map to check to see if we've managed to capture exactly what we see in the biology. And that allows us to start getting the specific color map for every type of cell. And it is different and it is unique, and it depends exactly on what are the different source cells. So this is how we structurally and functionally start connecting the neurons, but now we have to turn these connections into functional connections. They have to be able to communicate electrically. So over the past 10 years, what